today we have Jean-Marc Goaillard. Without further ado, I give microphone to Michael Zbili, who will introduce our speaker, and we proceed with today's talk. Hope you will enjoy it.
Hello, everyone. So thank you, Jean-Marc, for coming and giving this talk about variability and covariation in ion channel expression, and also variation in electrophysiological properties. So to introduce a bit Jean-Marc, he started to interest in the variability among neuronal networks and neuronal properties while doing his postdoctorate in Yves Marder's lab in Boston, where he studied variability of channel expression in stomastogastric neuron of the crabs. And he also was interested in the covariation in channel expression in those neurons, invertebrate neurons. And then he started his team in Marseille, where I met him during my PhD, and where he applied those concepts to midbrain dopaminergic neurons. And in this team, they did quite a huge work by combining patch clamp recordings, computational modelings, single neuron transcriptomics, and information theory approach, trying to see how the variability and the covariation in ion channel expression can explain, or partly explain, we'll see, the variability in electrophysiological properties of neurons. So thank you, Jean-Marc, and I let you doing the presentation.
Okay. Thanks, Michael, and thanks, Maria and Alexi, for inviting me. Okay, so as Michael said, what I'm going to talk about is variability and covariation in ion channel expression and electrophysiology properties in neurons, and I'm going to be showing data from crustacean neurons and from midbrain dopaminergic neurons, because basically the finding we made on these two very different types of neurons are quite similar. So the basic question we're trying to understand is, you know, how do we understand the electrical phenotype of neurons? So in other words, are we currently able to quantitatively, meaning mathematically, explain how a given neuronal population produces a highly specific and reliable pattern of activity? And basically, in my opinion, our current understanding is quite poor. So the answer to this question is that we don't really understand today how a neuron produces a given pattern of activities. I'm going to give you some elements to understand that. So just as an introduction, we know that in the mammalian brain, for instance, we have many different types of neurons. And these different type of neurons here, do you see my cursor, by the way?
Yes. Okay.
So I take here three different iconic types of neurons. So the midbrain dopaminergic neuron, that's the one we're working on. The CA1 pyramidal neuron, you all know that, quite similar to the neocortical pyramidal neurons. And then the Purkinje neuron. And we know that these different types of neurons are distinguished by very different morphologies. For instance, the Purkinje neuron have a very complex and almost two-dimensional dendritic tree, while the midbrain dopaminergic neurons have a fairly simple dendritic tree with a dendrite arising axon. And we know that from the point of view of their activity, they have also quite different activity. So both midbrain dopaminergic neurons and Purkinje neurons are pacemaking neurons, meaning that they're able to sustain a regular pattern of activity in the absence of any synaptic input. However, the midbrain dopaminergic neuron fire at 1 to 3 hertz, whereas the Purkinje cells fire at 40 hertz. And this is the kind of phenotype you can see in vitro. And in vitro, obviously, a pyramidal neuron is not fed by any synaptic input. So except if you inject a small step of depolarizing current, it won't emit spontaneously action potentials. So we know that all these different types of neurons have these different patterns of activity. And these phenotypes, electrical phenotypes, are in some way stable because you can record in any country, if you take a rat or a mouse that is mature, you will find more or less the same phenotype, you know. So the phenotype is pretty stable. And the phenotype is different between the cell type, not just because of the spontaneous pattern of activity, but you can go further and try to define more specific properties like the shape of the action potential. And there you see again differences. The midbrain dopaminergic neurons have a fairly low amplitude action potential, but fairly broad, whereas the action potential in the pyramidal neurons has a larger amplitude and is thinner. And if you go to Purkinje neuron, then you have a sub-millisecond action potential, super fast action potential, typical of usually of GABAergic fast spiking cells. So we know that electrical phenotypes are stable across animals and over time, because, you know, if you record a Purkinje neuron from a 3-week-old animal or from a 1-year-old animal, you will find pretty much the same activity. So what do we know about this electrical phenotype? Well, I'm gonna now focus a little bit more on the midbrain dopaminergic neurons. So these neurons are neurons from the substantia nigra pars compacta or from the ventral tegmental area, but in our case, we focus more on the substantia nigra. And these neurons project either to the striatum or to limbic area or cortical areas for the VTA neurons. And we know that these neurons in vitro in a slice preparation will generate this pacemaking activity, so tonic regular pattern of activity. And we have a pretty good biophysical knowledge of the channels that are responsible for this activity. So obviously there are intrinsic conductances because we're here recording in the absence of synaptic input. And we know that the HCN channels will play a role, that the KV4 channels, the A-type potassium current will play a role, that the transient sodium channels obviously are important, not only for the spike, but also for the depolarizing phase before the spike. Then we have L-type calcium channels that are involved, T-type calcium channels also, and calcium-activated potassium channels. So we have fairly good biophysical knowledge. So the second step of knowledge about the phenotypes is that we know that they are produced by well-identified ion channels. And in most neuronal types in the mammalian brain, we know pretty much the subtypes of ion channels that are producing the electrical phenotype. Then we also have a pretty good knowledge of the biophysical properties of these different channels. So here you have the typical waveform of a midbrain dopaminergic neurons, and here you have the main activation curves on the right here, plane lines, and inactivation curves with the grid lines for the main outward current and the main inward current that are expressed by midbrain dopaminergic neurons. And so it's basically here sigmoidal curves that represents the voltage dependence of these different conductances that you can find in dopaminergic neurons. So we have also a good description of the biophysical properties of these ion conductances or the ion channels for most channel subtypes in most neuronal population. And then we know also we have a pretty good description of the functional influence of these different types of channels. So here I'm going to show some pharmacological results from different groups showing the specific influence of KV4 channels. So in midbrain dopaminergic neurons, every time that you block specifically the KV4 channels with the scorpion toxin AMMTX3, you induce an increase in the spiking frequency. You also induce an increase in the spiking frequency if you block the HCN channels responsible for the IH current. Then if you block the CAV 1.3 L-type calcium channel, you get robust decrease in the firing rate. And if you block the SK channels, then you don't change the frequency, but you increase the irregularity of the frequency. So instead of having a very regular pattern of activity like you see here, you get a more irregular pattern of activity that will have a coefficient of variation that is in the higher values. So we know pretty much the functional influence of each ion channel type, and we have a pretty good description, once again, depending on the cell type. So the question is whether this knowledge is sufficient to explain how neurons invariably achieve their expected electrical phenotype and maintain it over time. And that's where I argue that this is not really sufficient to understand how this happens. Why? Well, the first reason why is because whenever we consider electrophysiological phenotype, we have in mind some kind of average behavior, some kind of typical or canonical behavior of a given neuronal population. But this idea of a mean as representing the behavior of a population might not be the right way of looking at the electrical phenotype. So considering just the mean is subconsciously considering that electrophysiological phenotype is invariant in a given neuronal population. I don't think that it's true. So I'm going to show you some data about that. This is data from dopaminergic neurons in the rat. And this is just a picture of the classical pacemaking behavior with the mean frequency of firing, but you can see that the standard deviation is quite big, okay? And the same if you look at the half-width of the action potential, the mean half-width is 1.56 millisecond, but the standard deviation is 0.38, meaning that pretty much the spike varies between 0.6 or 0.7 millisecond and 2 milliseconds. So you have a threefold variation. And you can see these significant variability here in the plots of the distribution of the log of the interspike interval, or log here of the half-width of the action potential, and a large number of recordings. You can see that there is quite a high degree of variability. And you can find the same exactly in the mice. So here I'm going to show recordings that come not only from wild-type mice, but also from Kv4.3 knockout mice. So these mice have a specific deletion of the Kv4.3 channel that underlie the A-type current in these neurons. And you can see again, maybe you should focus on the gray scatter plots that correspond to the wild-type. You can see that there is a large degree of variability. You go from 300 millisecond to almost 1500 millisecond for the ISI, meaning that you have almost a 5-fold range for the interspike interval. The coefficient of variation is also quite variable. And if you look at the post-inhibitory rebound, which is an electrophysiological signature of these neurons, you can see that it varies also quite a lot here on the right, from 200 millisecond to almost 1500 millisecond. You also can see, that there is some variability in the amplitude of the voltage sag, this signature here of the H-type current. And you find the same kind of variability for action potential properties. So you can see that the action potential threshold varies between -28 and -50 millivolt. Action potential amplitude varies pretty much between 50 and 80 millivolt. Action potential half-width, consistent with what I showed for the rats data, varies between 1 and more than 2 milliseconds. And the amplitude of the after hyperpolarization, so the voltage of hyperpolarization here after the action potential, varies between -65 and -85 millivolt. So every single electrophysiological parameter displays considerable cell-to-cell variability, even when we are in a well-identified neuronal population. Here I'm just talking about substantia nigra dopaminergic neurons that we specifically recalled from by localizing them anatomically in the slice, in the midbrain slice. And then we have a few electrophysiological parameters, such as the pacemaking frequency, the width of the action potential, and the presence of these biphasic post-inhibitory rebound to ensure that what we're recording from our dopaminergic neurons. And then we can do, obviously staining of dopamine synthesis enzyme to confirm that we're recording from dopaminergic neurons. So even in a well-identified neuronal population, every single electrophysiological parameter displays considerable cell-to-cell variability. Now, if we go back to the kind of question we're trying to answer, how do we go and how do we predict in a reliable manner the phenotype from the values of the properties of ion channels, we need to ask the other question, which is, are ion channel properties invariant in a given neuronal population? And again, not really. So having in mind a mean behavior, a mean value for ion channel properties is probably not the right way of looking at the picture. So I'm going to show you first data from the crab that I obtained when I did my postdoc in Yves Marder's lab. So the crab is Cancer borealis, and we studied specifically the stomatogastric nervous system, which is a small ganglion controlling the contraction of the stomach. And basically, the only thing you need to know is that this network can be simplified to these different cell types. So the PD, pyloric dilator, LP, lateral pyloric, and PY, which are the pyloric neurons. And it's basically the interconnection, the electrical coupling, and the inhibitory connections between these neurons that will generate a triphasic rhythm where you have first the pacemaker kernel firing, so the PD neuron, and then you have the LP neuron firing here, and then you have the PY neuron, and then the cycle starts again, PD, LP, PY, PD, LP, PY, PD, LP, PY. Okay, so we have these three different types of neurons. They are interconnected by inhibitory connection. They generate a triphasic rhythm. And so with Dave Schultz, we started analyzing the properties of these neurons, and in the first study we focused on the lateral pyloric neuron, so the LP neuron. And you can see here that you have two LP neurons which have virtually identical patterns of activity when you superimpose without scaling the time. When you superimpose the voltage waveform, you can see that it's very similar. So these two LP neurons have been recorded from two different animals. And then what we did in these same neurons is that we measured the conductances of three different types of potassium channels, the delay rectifier, the calcium activated potassium channels, and the A-type potassium channels. And we found quite a big range of variability in the values of conductances of these three different currents. And interestingly, when we measured by quantitative PCR, when we measured the expression levels of the corresponding ion channel genes, so SHAB, BKKC, and SHAL, we found that there was also quite a lot of variability at the mRNA level. And on top of that, we found that for the two channels that were the most variable, BKKC and SHAL, the values and the variation in expression was correlated with a variation in conductance. So we found that ion channels displayed significant cell-to-cell variability and in conductance and in expression, even in neurons with virtually identical activity patterns. Then later, we got interested in looking at the IA and IH current in rat neurons, in the midbrain dopaminergic neurons, and we started analyzing the properties of these two different currents. And you can see here two different dopaminergic neurons, which in fact have quite similar firing frequency. And you can see the H-type current here that is quite larger in the cell 1 compared to the cell 2, whereas the A-type current is quite larger in the cell 2 compared to the cell 1, okay? So we repeated these kind of measurements in many neurons, like more than 18 neurons, and we found that the amplitude of IH was quite variable, going almost from 0.5 to 2 nanoamps, so a fourfold range. The variability in IA amplitude measured at -40 millivolts was even more variable, probably also affected by the variation in voltage dependence. And the variation in voltage dependence also was quite strong from cell to cell. So even what I showed you before, sorry for this, but this idea that we can you know, have the intuition of the activation curve of IH sitting here in this range of voltage, we shouldn't see it that way. We should see it as going from there to there, basically, and considering a range instead of considering a mean value. And then you can also look at the expression level of channels. So we did that more recently by using microfluidic quantitative PCR on mouse midbrain dopaminergic neurons. And you have here the level of expression of the Kv4.3, so the channel that is responsible for this current, and the SK3 channel, which is a calcium activated potassium channel that controls the regularity of firing. And you can see again that there is quite a large variation in expression for the expression of these two ion channels, even though there are clear also differences in the level of expression between midbrain dopaminergic neurons and neighboring non-dopaminergic neurons. So we have, again, a large cell-to-cell variability in expression and in function of ion channels. And unsurprisingly, these induces large variation in the functional influence of ion channels. So if we go back to the plots that I showed you before that show the effect of these 4 different types of ion channels on firing, you can see that even though the average behavior is always in the same direction, there are some cells, for instance, that are very sensitive, very sensitive to the blockade of Kv4 channels, and some other cells are much less sensitive to the blockade of this ion channel. And this is true for HCN channels. This is true for CAV1.3 calcium channels. You can see here that this cell is almost undisturbed by the blockade of CAV1.3 channels, whereas in this one, firing is abolished by the blockade of this type of ion channel. And it's the same for SK channels. So, so if we go back to the original question, now we need to rephrase the question. We have a significantly variable phenotype with, a range that is quite, a target phenotype that is quite stable from animal to animal, but with significant quantitative variation. On the other side, or on the lower level, we have ion channels with highly variable level of expression, highly variable biophysical properties. And how do we go from this variability in ion channel properties with the achievement of a specific phenotype? So I'm going to show you first what theory says about that and what computational studies have taught us about this relationship between variation in ion channels and variation in phenotype. So first of all, if we go back to the voltage-dependent properties of our intrinsic conductances, what you can see immediately here is that for any range of voltage, so let's say the hyperpolarized voltage that will define the phase, the depolarizing phase, but also the after hyperpolarization, after the action potential, you have several potassium channels and several inward currents that are activated in the same range. And this is true for pretty much every range of voltage, okay? So instead of thinking about one ion channel having one specific function, we should rather think about one voltage domain being defined systematically by several different ion currents or ion channels. So every voltage domain is determined by several ion channels or ion currents. And this has a direct consequences in terms of biophysical solution to generate the same waveform. And this has been demonstrated by several computational studies. It means that in theory, multiple solutions are available to generate the same waveform, okay? If you have several parameters that are involved in defining the same feature, the natural consequence is that this feature can be defined by multiple quantitative solution arrangement of values of these different parameters. So we actually investigated that point with Adam Taylor on the stomatogastric nervous system. So still on the lateral pyloric neuron. So this guy in the middle here of the pyloric network. And what Adam did is a realistic model with several compartments and with specific expression of different intrinsic conductances in the different compartments. And then he tested this model. He generated a database of models with different values for each of the conductancies and each of the properties of these conductancies. So he generated millions of models basically, and then he constrained the model to reproduce the behavior and the range of behavior that was observed for the LP neuron in all the crab recordings that I had obtained. And so you can see here that you have, for instance, here 6 different examples of LP neurons generating similar waveforms. So for instance, the red and the orange are generating very similar waveforms in terms of spiking and in terms of the amplitude of the voltage waveform also. But if you look at the distribution of values for the different conductances, you can see that they're all over the place, and there doesn't seem to be a strict or scaling relationship between the different conductances. So if we look more specifically at the red and orange models, you can see that there is no clear relationship that is maintained between the different biophysical parameters. So we can look at it differently, and we can look at the relationship that exists between the different biophysical parameters. And here, I mean, this table is obviously unreadable, but the only thing to remember about it is look at the squares that are blue or red, because they mean that there is a significant positive or negative correlation between these two parameters. And you see that the majority of the squares don't have any color, meaning that most of the biophysical parameters are not correlated to each other. And even when there is a positive or negative correlation, you can see that it's kind of a small correlation. So there doesn't seem to be any specific quantitative relationship needed between specific parameters to achieve the proper behavior of an LP neuron. So in other words, taking a picture from a review from Mother and Taylor, we can summarize these to having a shape of the parameter space that is basically without any specific orientation, okay? So if we consider the majority of white squares here that have no specific relationship, we would expect that if we were able to represent the whole parameter space in the whole number of dimensions, we would get something that is mostly spherical and without any without specific orientation. Okay, but there were other theoretical studies that explored similar issues. So for instance, Guillaume Drion in 2011 explored the problem of pacemaking in midbrain dopaminergic neurons. So you have here models of midbrain dopaminergic neurons. The black trace shows that all these 4 models are generating pacemaking behavior with similar frequencies, and these 4 different models are located in a same small region in parameter space. And then what Guillaume did is vary the density of the sodium current and the L-type calcium current, because both of these conductances have been shown by different studies to be involved in pacemaking. And what Guillaume was able to show is that, first of all, you have a very large parameter region of the parameter space where you can generate stable pacemaking. But then, depending on the region where you start from, removing the calcium current or removing the sodium current turns out to maintain pacemaking or abolish pacemaking, okay? Just because you're starting from a slightly different region, you end up in a region where the neuron is just hyperpolarized and not generating any pacemaking. So here, for instance, you see in the model 1 that blocking the calcium channel doesn't change the spiking, but blocking the sodium channel removes the pacemaking. In the neuron 3, blocking either conductance doesn't have any effect on the firing, whereas in the neuron 2, the blockade of either the calcium current or the sodium current abolishes the firing. And the neuron 4, it's only the blockade of calcium channels that abolishes the oscillations. So interestingly, what this model tells us is that the best relationship between these two conductances to maintain pacemaking is a negative relationship. So we would expect that, and in that case, we just have a bidimensional parameter space. We would expect that the best solution for maintaining pacemaking is to have a negative relationship between GNA and GCA, and a negative relationship that is parallel to the boundary between the pacemaking region and the hyperpolarizing region. So this theoretical study is telling us that there might be specific relationships that are needed between biophysical parameters to generate a stable phenotype. Then I'm just showing you the summary results of this study from Lem and Calabrese, which was a computational study performed on leech motor neurons. And what these authors found was that all the models that were generating the proper behavior for their specific type of motor neurons ended up delineating negative correlation between the conductances that had the same functional effect. So all potassium conductances were negatively correlated. Calcium and sodium conductances were negatively correlated. And then in addition, in all these models, the values between the antagonistic conductances, so the calcium or sodium, and the potassium conductances were positively correlated. So in this theoretical study, it turned out that antagonistic current had positive correlation and synergistic current had negative correlation. So this theoretical study told us that in some models, at least, specific relationship that depend on the functional overlap between the two conductances that are considered, specific relationships are needed or are coming out of the satisfying models. So based on these theoretical studies, we may imagine that specific quantitative relationships between underlying ion channel properties may explain how a specific phenotype is generated. But this is all theory. So what happens in biology? The first thing we did with Dave Schultz on crab neurons is to analyze the higher order relationship between the gene expression level in different cell types in the pyloric system. And I'm going to show here just two examples. The examples of the PD neuron, part of the pacemaker kernel, and the LP neuron that I showed you before. And we did that on 6 different types of neurons, and the take-home message was basically the same. What we found with Dave was that the levels of expression of different ion channels were strongly correlated in different cell types, but the set of ion channels involved in the correlations were different depending on the cell type. So for instance, in the PD neuron, we had correlations between BK-KCA, the sodium channel, HCN channels, shell channel, but also Dave showed in a different study between SHAB, CAV1 and CAV2. Whereas you can see that in LP neurons, the sodium channels and the BK-KCA are not correlated with the level of expression of the other channels. So we have here different modules of correlated ion channel that seem to be specific of a given neuronal type. And I don't know what I'm showing here. Oh, yeah. So in another study that I did after this one, I also analyzed the properties of the synaptic inputs received by the LP neurons, in particular the inhibitory synaptic input received from the PD neuron, and also the properties of the neuromodulator activated current received by the LP neuron. And what I found is that there was a positive relationship, a positive correlation between the strength of this inhibitory synapse received from the PD neuron and the strength of the amplitude of the neuromodulator activated ion current. So we also found some covariation in ion channel function, because in this case, this was all based on electrophysiological measurement and not on qPCR measurement of gene expression level. We found similar results in midbrain dopaminergic neurons. As I showed you before, we found that there was a lot of variability in the level of expression of ion channels. So this is for midbrain dopaminergic neurons in green and non-dopaminergic neuron in red. However, when we plot them in a 3-dimensional space, we can see that even though there is some significant overlap, the points are clustering in different regions. But more interestingly, what we found when focusing on the ion channels that are involved in defining the electrical phenotype, we found that there were specific correlations between these ion channels. So for instance, the SK3 and NAV1.2 ion channels were strongly correlated in their level of expression, whereas SK3 was not correlated with the expression of Kv2.1. NAV1.2 was correlated strongly with Kv4.3, but was not correlated with the level of expression of CAV1.2. And so we ended up with the following picture where we identify, similar to what we saw in crustacean neurons, specific modules of ion channels that were correlated in their level of expression. And if we reduce to the most significant correlation, you can see that you have a small module comprising NAV1.2, SK3, GIRC2, Kv4.3, and HCN2 that are correlated together. So that's a pairwise analysis, but as we discussed with Alexis, Maria, and Michael, sometimes you want to go beyond pairwise analysis, but as I'm kind of late, I will pass on that and I can come back to that. But basically, we know that these statistical dependencies go beyond the pairwise relationship. So what we identified in dopaminergic neurons is that not only were ion channels correlated in their level of expression, but they were also correlated with the level of expression of a module of genes that are involved in defining dopaminergic metabolism and signaling. So you can see that all the ion channels here in purple, a number of ion channels are correlated together, and they are also correlated with the genes that are involved in dopaminergic metabolism and signaling. So in terms of data, it gives you that kind of four-dimensional plot where you can see that there is a strong covariation in the level of expression of the ion channels. So for instance, GIRC2, Kv4.3, NAV1.2, and then the dopamine transporter. Okay, I'm gonna pass on that. So what about the function of ion channel? So I showed you before that there was quite a bit of variation in the properties of IH and IA in rat dopaminergic neurons. But what we found with Julien Mandola and Adele Woodhouse was that these variability in the properties of these two ion channels are in fact hiding a covariation in the voltage dependence of these two ion channels. So the voltage dependencies of IH and IA are not independent, they are actually correlated together, positively correlated. So the more hyperpolarized IA in a given neuron, and the more hyperpolarized the activation voltage of IH. And interestingly, very recently, we found that it was exactly the same in mouse neurons. And you see the correlation is pretty much the same. The range of voltage is very similar. And the strength of the correlation is also very similar. And to finish about this covariation in ion channel expression, I'm going to show some data from fast spiking vestibular nucleus neurons. So Kodama and colleagues did this study in 2020 where they analyzed the level of expression of 14 different ion channels in vestibular nucleus neurons. And these neurons were identified based on their projection pattern and therefore their functional implication. And they analyzed the relationship between the levels of expression of these different ion channels. And once again, they identified a module of interconnected ion channels that are strongly correlated with each other. You can see here that you have a module of 7 ion channels that are almost all-to-all connected in terms of levels of expression and correlation in levels of expression. And in their case, they thought that this module is in fact the module that allows fast spiking, because all the vestibular nucleus neurons they measure the expression of these ion channels from were all fast spiking vestibular neurons. So the conclusions from this part is that widespread covariation in expression and in function of ion channels are observed in neurons. That unlike the theoretical predictions, the only kind of correlation we find in expression levels are quasi-linear positive correlations. And I think this raises a lot of questions. And I won't go into the answers to these questions. We're currently thinking of how to understand this problem. And these quasi-linear positive correlation, which are a specific subset per neuronal type, define neuronal type specific ion channel co-expression modules. And we think that this has a real functional relevance for answering the question that we asked first: how do you generate a specific electrophysiological phenotype in a given neuronal type, and how do you maintain it over time? So now the question we can ask, we've been talking about variability in electrophysiological phenotype, variability in ion channel expression. We showed that there were covariation in ion channel expression and ion channel properties. Question is whether electrical phenotype can also be defined by modules of statistically dependent parameters. So we started being interested in correctly defining the electrical phenotype of dopaminergic neurons like almost 10 years ago with Marcel Dufour, a former PhD student. And we did a study in rat midbrain dopaminergic neurons. And instead of having strong hypotheses about what may be relevant for the function of the neurons, we decided to have an unbiased or as little biased as possible investigation of the electrical phenotype. And we decided from each neuron to measure as many electrophysiological parameters as we could, and to say, okay, at least if we're measuring all the electrophysiological parameters, we won't miss anything about the phenotype. And in that way, we decided to not make strong assumption about which electrophysiological parameter might be critical or irrelevant to the behavior of the cell. So we measured the pacemaking behavior, so by measuring the interspike interval, but also the coefficient of variation. Then we measure the passive properties, input resistance, capacitance, time constant of the membrane. And then we measure the post-inhibitory rebound properties with the rebound delay, the voltage sag, then all the properties defining the shape of the action potential, and then also the excitability properties. How does the neuron fire at the beginning of a depolarizing step, at the end of a depolarizing step? What is the accommodation profile of the neuron, and so on? So we ended up characterizing the phenotype by measuring 16 to 24 electrophysiological parameters. And we did that over time because our goal was to determine when the electrophysiological phenotype of in vitro midbrain dopaminergic neurons was reaching stability. When could we, what was the age at which we could consider that a neuron was mature from the electrophysiological point of view. So we investigated 15 different developmental stages. And so as an example, you can see here the result for the pacemaking activity. You can see that newborn neurons, neurons from newborn animals are mainly bursting with a very high coefficient of variation for the ISI. And then neurons become irregular, and finally they become regular pacemakers, which is always considered to be the mature behavior. So we extracted, values for that, depicted the developmental profile for each parameter, and we ended up with like this kind of graphs where you have a lot of box and whisker plot, a lot of lines, and the idea was to get some global visualization of all these variations. So we turned to PCA, and reducing to 8 electrophysiological parameters, we could get this nice developmental trajectory of the electrophysiological phenotype in principal component space, where you have the very young neurons here, P2, P3, and then the neurons jump to a first region where they stay between more or less P5 and P10, and then they jump to a region where they will reach stable mature behavior. So they're all associated with different pattern of activity, here bursting, here irregular, here regular. But most interestingly, what we found, if we look more precisely at the mature stages of these neurons, we found that the relationship between the PC1 and the PC2 was not random. So you can see that there is a strong correlation at each developmental stage. There is a strong correlation between the value of PC1 and the value of PC2, okay? Suggesting that this stable phenotype is not just a random walk around a given mean value, but that it relies on specific relationship between the underlying electrophysiological parameters. So we could go back to the parameters and analyze the statistical dependencies. So similar to what we did on the gene expression level. And you can see here that you have a lot of correlations. Some of them are completely evident and related to how we measure parameters, but some of them are less obvious. So we did also that recently on mice neurons, and in particular, we compared the phenotype between wild type mice and Kv4.3 mice. And you can see that again, you have this modular organization. So we restricted here to, I don't know how many parameters, maybe 12. And you can see that you have a modular structure, and this modular structure corresponding to correlation between different electrophysiological parameters changes between the wild type mice and the mice where the Kv4.3 channel was deleted. So here, all the common correlation are in black between the two strains, and the red correlation are the ones that are present in one strain, but absent in the other. So you can see, for instance, that AHP and start frequency are correlated in the wild type, but they're not correlated anymore in the Kv4.3 knockout. Whereas in the knockout, the start frequency is correlated with the input resistance, and it's not correlated with the input resistance in the wild type. So the conclusion of this second part is that electrophysiological parameters also share correlations, or more generally, statistical dependence, that define electrophysiological modules. And these modules can be modified by chronic perturbation that modify ion channel expression. Well, actually, on these same experiments, on these same neurons, we have done not only the electrophysiological recordings, but also single neuron transcriptomic analysis using microfluidic quantitative PCR. And so we did some analysis of the modules of expression. So you have the same genes that I showed before, but also some additional genes. And once again, here we're showing the significant correlation by the links between nodes. The black ones are maintained between the wild type and the Kv4.3 knockout. The red one are the ones that are specific of one given strain. And you can see that some of the genes are specifically correlated in the wild type and not correlated anymore in the Kv4.3 knockout. So the general conclusions of these talks are that ion channels are organized as co-expression modules, that these modules are modified after chronic ion channel perturbation, and that electrophysiological phenotype is also made of modules of parameters that are modified by perturbations. So the ultimate goal of all this work, and I think that the general conclusion of this work, is that instead of thinking about one ion channel affecting one specific electrophysiological feature, the way we should understand the relationship between ion channels and phenotype is more of the following, where we have one genetic scales, where we have the ion channels with specific modules of covariant ion channels or other type of statistical dependencies in the expression of ion channels. Then at the other end, at the very top, we have the phenotypic scale, where the phenotype is made of different electrophysiological parameters that also share specific statistical dependencies that may be specific of the neuronal type we're interested in. And the question is, how do you go, from one to the other? So we haven't started addressing this question yet, but we have some elements. Actually, we did it in the stomatogastric nervous system, and we got some nice results, although we didn't understand them. But there were some studies that tried to really understand how you go from this bottom scale to this top scale. And then there were other studies, in particular the studies from Timone that he performed in the mother lab, some really interesting modeling studies showing how variation in electrophysiological phenotype can regulate the expression of ion channels. Interestingly, these studies from Timone showed that with very simple homeostatic rules that modify the expression level of ion channels depending on calcium level, what arises from this regulation are strong covariation in the levels of expression of ion channel, just like what we found all the neuronal types that have been investigated for this problem so far. Okay, so I think I'm done pretty much with presenting you the data. Now, obviously, there are a lot of pending questions raised by these results. The first one is to determine whether this covariation of ion channel expression really explain the stability of electrical phenotype. And so far, we haven't gone that far. We haven't performed the perturbation experiment that could demonstrate this point. So this question is still more or less unanswered. We have partial answers, for specific pairs of ion channels, but it's only very partial answers. Then the second question is, what are the genetic mechanisms that are responsible for the emergence of co-expression modules? And again, we don't really have the answer. And a related question related to that one is whether these co-expression modules are part of homeostatic feedback loops. And that would basically make the link between the first two questions. If you have genetic regulatory mechanisms that are, for instance, dependent on activity level, then that would mean that you have genetic mechanisms responsible for co-expression modules, and that these genetic mechanisms are directly responsible for the stability of the electrical phenotype. So with that, I'm done. And so I need to thank all the people who were involved in this work. So Alexi Ajeri did part of the recordings of the wild type mice and K4.3 mice. But a lot of that was done by Monica Tapia, who did also the single neuron transcriptomics studies, both with the patch clamp and without the patch clamp. Fabien Tel was involved in a lot of the analysis of the data. And Christine Formizano was involved in the transcriptomic analysis, and Pierre Baudot, generated developed the mutual information analysis that I didn't have time to present yet. Thank you very much.